Hello, welcome to this web class. Just give me a quick thumbs up or something if you guys can hear me okay, just to check. Never sure if it started properly. Thanks, Evan. You can hear me, good. It looks like it's just a spinning wheel though. Can you see me as well? Cool. Perfect. Thanks. How is everyone this evening? What have you been up to this last week? Rhea, some work experience. Very nice. What are you getting experience in? What field of work? Yeah, I'm very good, thanks, Khadija. I'm in a good mood. Those of you who are dialing in from the UK will probably be aware we had a presentation, like a briefing from the Prime Minister yesterday, sort of outlining when we're going to be able to do things again. And it was kind of like earlier than I expected. Like, should be no restrictions by June, which sounds great. Rhea, Morgan Sindel Construction, designing a hotel. Nice. So you want to go into like civil engineering, or are you more in like the design, interior design side? Nima, do I actually like biology? No, I'm just forced to be here each week. No, I do like biology. And when did I know I wanted to do it for A-level? I feel like definitely by the time I was doing my GCSEs, I was like very keen on biology. I really like geography as well. So I knew I definitely wanted to do biology and geography. They're my two definite ones. And then I also picked chemistry and PE as well. Back when we used to do like, you did 4 for AS and pretty much everyone dropped the fourth AS for A2 and then just 3 for A2. But I think now it's quite common just to do 3 the whole way through. Yes, Alicia. Wait, sorry, Alicia. Am I saying that correctly? I was a teacher for a few years in a school in London, but now doing like online tutoring and other tutoring things, basically. My degree was just in biology, and most of the way through a master's in conservation. Alicia, okay. Cool, I'll try to remember that. Alicia, oh yeah, I see it now. I'm never very good at like reading a name and then working out how to pronounce it. Yes, this is for AQA. This is for all the exam boards basically. So it doesn't matter which GCSE exam board you're doing, this will be relevant. Cool, right, so I just waited a couple of minutes to give everyone a chance to dial in. I think that's enough time now. So let's make a start. I'm just going to share my screen. Perfect. So yeah, hopefully you're seeing a PowerPoint now. Is it specified for AQA? No, it's for all exam boards basically. If there's some things that aren't relevant for AQA, I would usually like point out, or for any of the exam boards. So if it's like most of the specs learn the same stuff, and that's the stuff we focus on in these lessons, so they cater for everybody. There are some deviations, and when those do come up, we'll let you know, so you don't have to like learn other specs content, basically. Music, no, unfortunately it's not A-level, so if you're joining for an A-level session, this might be quite easy for you. You do have to learn mitosis again at A-level. Oops. So this bit would be relevant, but it's much more basic what you cover at GCSE. So yeah, you probably wouldn't get too much out of it if you're on A-level level. Anyway, let's dive in. A little bit about me. I've drawn over my face. So my name's Alex. I'm the head of biology here at SnapRevise. I've been teaching and tutoring, as I mentioned already, for 4 or 5 years now. So let me talk about SnapRevise. So this is a like learning revision website. So I just realized there's like the eraser thing is like stuck on the screen, but it doesn't show on my screen, but it shows on the YouTube screen, which is really weird. Let's just put it over here. No, doesn't want to move. Okay, yeah, put it behind my picture now. So SnapRevise, really useful revision and learning tool. We've been running for A-level for a few years and now launching a GCSE package. So that's why we're doing these YouTube classes. So we'll be running 4 classes a week for each subject to start off with, and maybe expanding that as well in the future. We go through all the content for all the specs. Got some great revision notes for you, sort of like cheat sheets, condensed summary packs, so you can download those. And all the sessions will be recorded. So if you ever miss one, or if you want to join in the future, all the ones will be available online. And you can also request one-to-one help with a tutor. So if you've got some issues and you've watched the videos and you've gone to a class and you're still not quite sure about a certain thing, you can ask for help directly from a teacher. So really, really useful package. Oh, a few people thought it was A-level. Yeah, by all means, you're very welcome to stick with it. It'll be like a little bit of revision, but it's definitely like at a GCSE level rather than an A-level level. So you might find it quite easy, but I don't want to put you off. You're very welcome to stay. But yeah. Okay, let's dive through. So we're running a competition. This is the last week that it's running. We did extend it a little bit at some point. So it ends on the 25th of February. Is that like next week or something? No. I never know what date it is now. Oh, 2 days time. So you've got 2 days to enter with a chance to win it. We're giving away 10 accounts. So well worth entering. Usually only give away 1 account. So then it's much less likely to win. This time, 10 accounts. So a good chance of getting in. Can you enter more than once? I don't know. Maybe. I don't know if it would be counted more than once. So it might be a wasting your time. Yeah, I'd probably say just better to put one submission in, but potentially you could manipulate it a little bit. Nice, good to hear. Yeah, everyone sign up. Super easy, and a chance to win a free account for the rest of the academic year. So I actually thought it was just for 1 month. So this is an even better prize than I previously realized. So the link is here, and it's also just been put in the chat. So yeah, sign up. Okay, that's the background. That's the sort of introduction. Let's go through the content for the lesson today. So we're going to cover the role of chromosomes in the cell cycle. We're going to look at the different stages of the cell cycle, basically the stages of mitosis, well, mostly focusing on mitosis. And then we'll look at why mitosis is important. We might not get through everything, but we'll do as much as we can. So these are the spec points. Like I said, it's for all specs. So sometimes talk about this a little bit more. Just going to skip over them today because I want to get to the content. But basically, this stuff we're learning is on all of the specs. So OCR, oops, skipping, EdXL. Also, if you're doing one of the international GCSEs, this is relevant for international IGCSE from EdXL or what's the other one? Cambridge. So yeah, it's all useful stuff. So a little recap. This is what we looked at in the last few sessions, or in the last week's lesson specifically. So what is the process that creates specialized cells? Nice. Yes. Differentiation. Yeah. So sometimes people call it specialization. Better to call it differentiation. Creating these different cells with different features, enabling them to be more efficient. So cells specializing means that multicellular organisms can be more efficient. Being a jack of all trades as a cell is not very good for performing your function quickly. So kind of like with people as well. If you want something done, you probably get in a specialist. If you want to build a kitchen, you get in a kitchen specialist. If you want to, that was a terrible example. If you want to sort your accounts out, you get in an accountant. If you want to put a new roof on your house, you get in a, I don't know what they're called, roofing person, roofer. You don't just get a bloke. You want specialists, and it's the same with cells. And that allows multicellular organisms to be way more efficient than like prokaryotes. And we can like, we've done a lot more than prokaryotes. They've stuck around as single-celled, and multicellular organisms can walk and talk and do all these fun things. So let's look at sex cells in particular, because they're highly specialized. So what are the two types of sex cell we've got? Yeah, nice, sperm and egg. So what are some features of both of those cells? Yes, nice. Many mitochondria. This is because sperm cells are very active. So they need mitochondria for ATP, which they use for energy. So to swim, basically. I can't remember the exact comparison. I think relative to their size, the distance they swim is the same as like swimming for a human to swim the Atlantic, which is like an insanely long way. People like struggle to swim the Channel, which is 20 something miles or kilometers. The Atlantic's like several thousand kilometers. So it's a very long way. I actually don't think anyone swum the Atlantic yet. I remember reading a few years ago about someone who was trying, but I don't know if they completed it. So yeah, they are good at swimming. Yep, so they're streamlined. And they have a flagellum, which helps them to swim. Oops, flagellum, like that with a U. I can't see it anyway. Has anybody mentioned? Yeah, the mid piece is where the mitochondria are. Very good. Oh, nice, haploid. Good. So they're actually both haploid. Maybe you meant that. So this is really relevant to the lesson today, because we're looking at chromosomes. So haploid means they have a half set of DNA. Ultimate revision, nice. That's what I was going to put up here, the acrosome. This is in the head, the very tip of the head. So just draw a sperm cell quickly. That's the flagellum. This is the many mitochondria, would be in the mid piece. And then the DNA is in the nucleus. And then at the very tip is this acrosome. And this contains enzymes. Those enzymes are used to digest the outside of the egg so they can enter the egg. The boss, oh, someone has done it. Benoit Lem, I can't do French names. Lecomte. Cool. Good to know. When was that? When did he make this crossing? I think that's a male name. I'm not sure though. What about egg cells then? Anyone got any features of an egg cell? Nice. Yeah. So jelly coating, or just like, yeah, that's fine. Coating. There's nutrients in there. Ultimate revision. Very nearly cracked a inappropriate joke at your comment. Becomes hard after fertilization. It's the opposite to the to the male. Okay, I did say it in the end. I tried not to, but I couldn't help myself. Nutrients, so we call that the stored in the yolk. And then what else do we say? Oh, the surface. Yeah, once one sperm has gone in, the outside, like, yeah, goes hard. It becomes impermeable. So this is really important because it means that only one sperm can get in. If more than one get in, this causes issues because there's going to be the wrong number of chromosomes. So what can we say? Oh, this is the granules are involved with this. So there's lots of little vesicles near the edge called granules. Make membrane or outside impermeable. Nice. So yeah, it's like a big race with the sperms. They're all racing, and only the first one to get in will succeed. Will be the one to fertilize that egg, and it's also the membrane changing structure. So yeah, it's not necessarily the first sperm to arrive. Normally, you can watch. There's like videos of fertilization because they do it in the lab for in vitro fertilization, so they can film it, and it's quite cool. So you'll see like the egg cell. Egg cell is way bigger than the sperm cells. Yeah, the largest cells actually. In fact, yeah, nice, exactly. So, and they'll be surrounded by like sometimes like many hundreds, all trying to get in, and only the first one in is the victor. So you guys are all winners. Every single one of you is the protege of a winning sperm, one of many millions. So if you're having a bad day, just remember that you're a winner. Yeah, they're quite short-lived sperms. Yeah, it depends. Like, yeah, somewhere between 3 and 9 days sounds about right. It depends on the conditions, how long they'll last. You can freeze sperm and they'll last like indefinitely. Cool. That's probably enough about sperm and eggs. Let's move on. So let's look at chromosomes. So what have we got here? We're sort of working our way up in decreasing size. So what are these things we're looking at? 3 and 4. Can haploid nucleus be 1? What do you mean by that? Oh, there's a DNA emoji. Didn't know that. Yes, that's good use of an emoji. Yeah, so this is DNA, which is a double helix. Correct. Then yeah, nice. So this is the nucleus. Contains many chromosomes. This is a chromosome, good. And this is number 4, anyone got it? Oh, nice, yeah, a gene. So DNA is located in the nucleus. It's stored as chromosomes, which are basically sections of DNA. So imagine all your DNA is one long strand like that. It's broken up into sections. We call those sections chromosomes, and we've got 46 in humans, or 23 pairs. I know I haven't drawn anywhere near 46 lines, but just to illustrate the point. So that's a chromosome. It's a long section of DNA. It contains many genes. So a gene is a smaller section of a gene. And basically, you can think of a gene being something that codes for a protein. So a gene contains the instructions to make one protein. So yeah, we've got lots of genes because we need to be able to make lots of different proteins. Nice. Yeah, Chromatids, a little bit different. Chromatid is like 1/2, so these are called sister chromatids. Maybe we're jumping ahead a bit here. Actually, when you see a chromosome like that in this X shape, it actually means it's actually two identical copies of the same chromosome. Chromosomes are actually just like would be 1/2. When they're in this X shape, it's because the cell's about to undergo cell division. That's the reason that's why they condense, and they have replicated before that. So actually, yes, this is two copies of the same chromosome, but we're used to seeing them as X's because they only form these like shapes when they're undergoing division. So that's why we draw them like that quite often. Prokaryote, yeah, this is a prokaryote, aka basically like bacteria. You can think of them as. This is just a DNA loop. So they don't have a nucleus. They have circular DNA. So this was like a eukaryotic DNA. If you straightened it out, it has a start and an end. Prokaryote DNA, if you straightened it out, like unraveled it, it would form a circle. So it doesn't have a start and an end. Yeah, these videos will be on YouTube for forever, I think, well, as long as YouTube exists. These are plasmids. So plasmids contain one or a few genes, and the prokaryotes might have many of them, and all the other genes are stored in the DNA loop. So sort of like be made up of sections. Each of these sections will be genes. Prokaryotes in general have a lot fewer genes than eukaryotes. They don't need to be able to make as many different proteins because they don't do as many things. Yes, exactly. People answered your question there. It's just a small loop of DNA containing a few genes. Bacteria actually can swap genes with other bacteria, which is a really cool skill that they have. So we obviously give genes to our children and we inherit them from our parents. So we can pass genes down generations, but we can't share genes with like our peers or our other adults. But bacteria can do that. So yeah, it would be a weird, well, it'd be a very different world if you could like, if you liked somebody's hair color, you could like get the genes for it and you could have that color hair. I mean, you can do that with hair dye, I guess, but it would be actually like in your DNA. That's what they do. Okay. So next up, let's look at chromosomes a little bit more closely. So chromosomes are inherited from your parents in reproduction. You get half from your father, half from your mother. In sperm, from mother in egg, or you can call an egg an ovum. So we can call this paternal DNA or paternal chromosomes and maternal DNA. Okay, let's just recap the difference between sex cells and normal body cells. So human body cell, sex cell, aka gametes. So actually, we should be using the word gametes whenever possible. So I was reading these comments, just catching up because I was looking down for a little while. Cool, nice, yeah, lots of good info on there. Yes, these are diploid and these are haploid. So what this means is they have a full set of DNA in a normal body cell and they have a half set of DNA in a sex cell. So actual numbers of chromosomes, there are 23 pairs. aka 46, whereas sex cells just have 23, not pairs. This is useful because when we get two sex cells, we get our sperm and an egg. We have 23 from the sperm plus 23 from the egg. This takes us back to I was trying to say 46 and I have just written 44 while I was saying it. That is not good maths. So 46 chromosomes, which is back to our diploid normal number. So it's really important. If we didn't form gametes that were haploid, every time there was fertilization, we would be doubling the number of chromosomes, which would be ridiculous. It would be an exponential growth in chromosome numbers, which obviously isn't feasible. So we form haploid cells. This is only an issue for sexual reproduction. If you reproduce asexually, like a spider plant, then it doesn't matter. Ali, what was your question? Why does animal cell have a vacuole? Okay, good question. Yeah. So only plant cells have large permanent vacuoles. That's what you need to say. There can be vacuoles in animal cells, but they are not a permanent feature and they're much smaller. So yeah, that's the issue. In general, they kind of just mark it for GCSE, just kind of think of like animal cells not having vacuoles. That's probably easier, even though they do technically, they don't have large permanent vacuoles, basically. In the loop of DNA, yes, Evan, it's called the nucleoid. You can also call it that. Yeah, that's actually the better name. Yes, basically a vacuole does help prevent a plant cell from like collapsing. Exactly. When it's got enough water, so a plant cell, the vacuole's in the middle. When it's got enough water, the vacuole swells up and it's exerting like pressure outwards. This pressure outwards, and then there's a cell wall around the outside of the plant that resists this pressure, and that creates a turgid cell. Not a very nice word, but that's the correct one. So yeah, it's turgid, and it keeps plants, it's like provides them structural strength. Exactly. So if plants are dehydrated, they wilt because the cells aren't turgid. Anyway, we're covering, it's all relevant. Well, it's all biology. It's all on the GCSE spec, but it's not what we're supposed to be doing today. So yeah, let's keep going through. So how are you guys feeling so far? Happy with that? Yeah, all of these topics you do in more detail at A level. Yeah. Because they're all like core biology stuff, you keep expanding on it in A level. Cool. Nice. 420. Great. So let's do a question. It's quite good to do like a bit of content and then a question just to check understanding and to see how that would be phrased in an exam. So some people sweat too much. Agreed. This is actually a medical condition they were looking at. I would say I put myself in that category, sweat way too much. But I don't have this disease. Oh no, genetic disorder. Maybe I do. I don't know. I've never been tested for it. That would explain a lot. So scientists have discovered hyperhidrosis is a genetic disorder. Gemma cells contain a gene that causes hyperhidrosis. Poor Gemma. Where about in the cells are genes found? Sweaty tryhards. Yes, in the DNA, exactly. So you could say in the chromosomes. Oh, sorry, for 2 marked, you need to say more than just in the DNA, obviously. So in the DNA, in the chromosomes. So 1 mark for that and 1 mark for that, basically. It was looking for chromosomes rather than nucleus, a bit more like specific. Can't. Oh no, sorry. You do get a mark for saying nucleus. I take that back. So you can also say in the nucleus. So yeah, if you put that, that's good. Nice. So hopefully that was easy enough. Let's do a few more like short answer questions. So border collies have 78 chromosomes in each body cell. How many chromosomes in a sperm cell from a border collie? Would a person have 2 genes that causes hyperhidrosis? That would depend if it's dominant or recessive, which again is another topic. But yeah, if it was recessive, then they would need to have 2 copies of the gene to show symptoms. If it was dominant, they would only need to have 1 copy of the gene to show symptoms. Yes, you guys are all on, you got the right answer, basically, yeah. You just divide that by 2, so 39. So because this is a haploid cell and the body cell is a diploid. Hope I haven't answered another question. Okay, how many chromosomes are in a fertilized egg from the border collie? Yeah, so fertilization takes our haploid gametes back up to the diploid number. So there's going to be 78. Nice. Finally, what substance are chromosomes made from? Nice. Made of DNA. Lady Prosecco. Are you old enough to drink Prosecco? Oh, I was about to write, I almost write down your name. I'm not gonna, yeah. I'm not gonna report you, don't worry. What substance is chromosomes made of? DNA. Nice. Final part, what part of the cell contains the chromosomes? Nucleus. Yeah, very good. Perfect. Happy with that? Let's move on. Yeah, nice. Lots of good answers coming in. Okay, let's look at the cell cycle and mitosis. So mitosis is like replicating a cell to create an identical copy of it, basically. The cell cycle is like, or mitosis is part of the cell cycle. So why do cells divide in the first place? What are DNA strips do the form of that? Lakifa, I don't fully understand your question, I'm afraid. Yeah, for growth and repair, basically. Growth of new cells, i.e. to get bigger, and repair of existing cells. Oh, well, sorry, like replacement, not repair. kind of overcomplicated that. Why do cells divide? Growth and repair. Think of that. And they repair to replace damaged cells. Just gonna write that because that's usually enough to score you 2 marks on a mark scheme. So I wouldn't worry about the rest. Where and what cells might be actively dividing in a human, let's say? What are some fast growing cells? Body cells need a bit more detail than that. Specialized cells, yeah, they become specialized. Bone marrow, yeah, lots of cells dividing in the bone marrow. That creates blood cells. Nice. Skin cells, yeah, they're not very long-lived. So they're always being replaced. That's actually, apparently that's what makes up most of the dust in a house. It's just like skin cells, so kind of gross to think about. But they're all like building up. Meristem in a plant, yeah, very good. An area of high growth in the shoots and the roots. Yeah, a tumor would be fast growing. Yeah, exactly. So cancerous cells. That's unwanted, but it is a fast growing cell. So some of the therapies to treat cancer, like chemotherapy, for example, the reason it makes like your hair fall out and makes you feel really sick is because it's targeting fast growing cells. So the tumor's fast growing, so it targets that, but it also targets all these other fast growing cells. So like hair cells, we should add on here. Hair follicles. Oops, spelled horribly wrong, whatever. And like fingernail cuticles, they're all fast growing, and the lining of your stomach, fast growing cells, and they get destroyed by the chemotherapy as well. So that's why you lose your hair. Sometimes you lose your fingernails, and you feel like generally really sick. Yeah. Yeah, with cancer, it's really good if you find it early, because the smaller the tumor, the easier it is to cut out and the less damage it will have done. And if it hasn't, the earlier you catch it, the less likely it's spread around the body. So it's called metastasis. Eventually, tumors will often spread to other parts of the body. Usually by the time that's happened, it's, well, not always, but it's quite often game over by that point. Like it's too hard to cut them all out now. It depends. People obviously do recover, but it certainly doesn't help your chances if it's spread to all parts of your body. Cool. So let's look at eukaryotic cell division. So when cells divide, produce genetically identical daughter cells, what do we call this? What not, I haven't got time to go too deep into cancer, I'm afraid. But I was looking at for one of my tutees, the like cancer charities websites are really good. They have lots of detailed information on that. We call it mitosis. Very good, yeah. Nice. So we're going from a diploid cell. And what are we forming in this case? 2 daughter cells, 2 genetically identical so these are all genetically identical, the daughter cells and the parent cell. Okay, we're split vote. 1 diploid, 1 haploid. 2 diploids, 2 haploids. Wow, it's really neck and neck. 3 votes for each. Oh no, 4 for diploid, 3 for haploid. Diploid has just clinched it. And yeah, well done, that is correct. So in this case, these aren't gametes. So we're forming diploid. diploid cells. It's only when we produce gametes that they're haploid, and that's meiosis. Yeah, very good. This is not sex cells, so they're going to be diploid. If you think about it, they have to be identical to the parent cell. Therefore, the parent cell had 46 chromosomes. These ones need to also have 46 if we're looking at humans. So yeah, mitosis is creating identical cells. Meiosis creates gametes. Really common to get those mixed up. Normal cell. You can remember because mitosis is happening in mitose. Don't know if that helps. Whereas meiosis is not. But yeah, it's really common to get those mixed up, and really easy marks that you're throwing away if you get those mixed up on an exam. Okay, so let's look at the cell cycle in a little bit more detail. So what are we looking at here? So the first stage, this is called interphase. What's happening in interphase? And this is stage one, sort of between here and here. This is like a normal cell, and this is a cell. Yeah, mitosis, IP mat is a good one to remember, which describes the stages, or IP more after T as an acronym for that, or no, not an acronym, whatever that's called. Mnemonic, I think. Yeah. So in interphase, the cell grows, DNA replicated. What else? Organelles replicated. Like that. Then stage 2 and 3, we've kind of, yeah, basically like preparing for mitosis, exactly. 2 and 3, we're looking at mitosis and cytokinesis. So mitosis actually means nuclear division, like dividing the DNA. Technically, that's what's going on. Plus cytokinesis. This is where the cell separates. So they start to like pinch off, kind of like putting a belt around a water balloon and like tightening it. It kind of looks a little bit like that. And eventually it gets narrower, 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 the waist in between. And eventually they pinch off and form two separate cells. That's cytokinesis. Metaphase is one of the stages of mitosis. So mitosis includes prophase, metaphase, anaphase, and telophase. They're all part of mitosis. I think, let me just check my notes. Technically only Edexcel actually need to know these different stages. The other specs, OCR and AQA, you actually don't need to be able to name them. You just need to be able to describe what's going on at each stage. But if you want to learn the names, that's no problem. So let's go through mitosis in a little bit more detail. So firstly, if you're Edexcel, or if you just know them, you can add what are the names for each of these stages? Yeah, potentially that was just for the triple awards as well, I think. So potentially if you're just doing single science, you don't even need to know the names for Edexcel either. Yes, the video is saved. Exactly. So the first one is prophase. Just if people are interested in the words. Second one is metaphase. Third one is anaphase. Fourth one is telophase. And then interphase. No, cytokinesis. So what's going on in each of those stages? If you're giving me an example, make sure you say like which number. No, you don't need the names for AQA, only for EdXL. But if you're going to go on to do biology for A level, then you might as well learn them because you need them for A level anyway. So what happens in prophase? Nuclear membrane disappears. Breaks down. And you can see that in this diagram. This is the nuclear membrane and it's disappeared between this and the next stage. Chromosomes condense. This means that they like shorten and thicken. Yes, supercoils is a good one. Exactly. Condensing, supercoil. Basically, so shorten plus thicken. You can only see DNA properly when it's condensed as chromosomes. If you look at a cell that's not undergoing mitosis, the DNA is all unraveled. You can't see it properly. The whole nucleus just looks like a dark shape. Yes, mitosis only happens in somatic cells. So somatic cells just means non-gametes, basically. All the other cells that aren't gametes are somatic cells. Exactly. Metaphase, yes, chromosomes line up in the middle. Middle of cell, we call that the equator, which is like kind of like a globe, same name. Sorry, I was looking at the comments. Callie, the names only for EdXL are necessary. Anaphase, these chromosomes are pulled to each end of the cell. This is one of those things that they're like, I kind of disagree with. Is you're being lied to, basically. Well, not lied to, but it's oversimplified. So you're gonna have to, unlearn it if you do A level. So they're actually the chromatids that are pulled to opposite poles. And some of you might be thinking, like, seeing that and thinking, like, yeah, I thought it was chromatids. You're right, it is chromatids. For GCSE, you can just say the chromosomes are separated, and yeah, won't go into them too much now. Body cells are diploid, yes, exactly. Yes, they're pulled apart by spindle fibers, exactly. And just like how the middle is called the equator, the ends are called poles. So it's based on the same as like a globe or a planet. Telophase. What happens in telophase then? So, yeah, DNA decondenses, or chromosomes, we say. Decondense, which is the opposite of condensing. Nuclear membrane reforms. Around there's two nuclei now. Nuclei, cytokinesis, separation of plus nuclei. Cool. Yeah. So it starts to split and eventually it does split. So this is called a cleavage furrow, and that gets like the join between them gets smaller and smaller and smaller. Eventually it pinches off. And we've got two new cells. We call new cells daughter cells. We call the original cell a parent cell. So our parent cell has become two daughter cells. And they're all, the parent cell and the two daughter cells are all genetically identical, unless there's been a mutation. But that's another story. Sickle cell anemia has one extra chromosome. No, sickle cell is not a chromosomal disorder. Sickle cell is caused by a faulty allele, but there are lots of chromosomal disorders. Like Down syndrome is one, Prader-Willi is another one, Turner syndrome. What's the one? What's his name? Katie Price's son, Harvey, has, I think he has Prader-Willi, like a chromosomal disorder. What does it say in part 5? Separation, oh yeah, I kind of scribbled all over that. Separation of cytoplasm and nuclei. Or the two nucleuses, basically. Cool. So, what next? How are you guys feeling with that? Making sense, more or less? Nice. Cool, good. That's what I like to hear. Let's do some more questions. So first question, can you explain why DNA replicates before the cell divides? Major difference in mitosis and meiosis, okay? Growth and repair. Two genetically daughter cells. Creates diploid cells. Meiosis for making gametes. They're haploid. And you actually create 4 genetically different cells each time. Unique cells. And yeah, meiosis is only for making gametes, basically. So the majority of the time, it's mitosis. Cool. So yes, lots of good answers there. So we need to replicate so that each ends up with a complete set. Ends up with a of chromosomes or DNA. Okay, nice. Cool, next question. A skin cell divides by mitosis once every day to produce new skin cells, and the old ones fall off and become dust. Calculate how many days it would take to produce 16 skin cells from one skin cell. So, what do we think? 16 days. Yeah. Nice. How did you calculate that? Oh, sorry. Not 16 days. We're trying to get to 16 skin cells. It's 4, yes. I was thinking about something else in my head. Yeah, we're trying to go from 1 cell to 16 cells. Each day, we double the number of cells. So day 1, we have 1 cell. Day 2, we have 2 cells. Day 3, we have 4 cells. Oh wait, no. Oh, in the first day, we go from 1 to 2. So I'm out by 1 here. So day 1, we go from 1 cell to 2 cells. Day 2, those 2 cells double again to become 4 cells. Day 3, 4 to 8. And day 4, 8 doubles to 16. So each time it's times 2 because they double. So 4 days. That making sense? Yeah, exactly. So you can sort of shortcut to work it out. It's 2 to the power of 4. That's equals our 16 days. So we could square root 16 as well if we wanted. How are we doing for time? Okay, we've got one more question. Let's quickly finish it off. Okay, mitosis occurs in plant cells during growth. Describe the division of a cell by mitosis. So we're looking for a 3 marker here, describing mitosis. So replicated are aligned on cell equator plus pulled apart. That's, if you're looking at the names, that's metaphase and anaphase. Cell divides by cytokinesis, producing 2 genetically identical diploid, let's throw that in for good luck, diploid daughter cells. Okay, so yeah, that would be 3 marks, like that. Cool, yeah, lots of good answers coming in there. Nice. So that's it. Hopefully you can now do all of these things, and you know about mitosis, you know about cell division, and you know about DNA and chromosomes. Do you have any questions on that? Yeah, they're cancelled, but you still have, you'll probably have to do something in your school. Exactly. Yeah, you're very welcome. No worries. Have a good day. Yeah, basically that's it. That's the end. So yeah, thanks for joining. Hopefully that was useful. And that was our last one actually. And if you like these sessions and you want to continue with them, then hopefully see you guys on our website. And we do these every week, like 4 a week for each subject. So this is actually out of date now. We're done. We might do occasional ones on YouTube, but if you want to join in regularly, you need to be migrating to our website. So don't forget, competition. A chance to win your free account, and yeah, hopefully see you guys in the classes in the future. Yeah, no worries. You're very welcome. Yeah, so hopefully see you guys in the classes. If not, on YouTube in the future. We usually do like one a month on YouTube. So we'll keep doing those, I think, but they won't be quite as regular. You submit the entries to here. Yeah, hopefully you guys all win, although 53 of you, guess you can't all win. Cool. Yeah, have a good evening. And don't forget, we've got a couple more classes. Shouldn't rush across them out. We've got 2 more classes this week. If you do GCSE maths or GCSE chemistry, join in with those classes. Let me just actually rub that out. Perfect. Have a good evening, and I will, yeah, see you soon, hopefully. Bye.